We are defined in large part by the genomes that we happen to come to the table with. So what does it mean when we can edit our own genomes? What does this have to do with viruses or copy pasting, or whether we are going to modify the story of our own species? This is Inner Cosmos, and I'm David Eagleman. I'm a neuroscientist and an author at Stanford, and in these episodes, we sail deeply into our 3-pound universe to uncover some of the most surprising aspects of our lives. Today's episode is about the remarkable situation we find ourselves in, which is that we now know how to read our biological inheritance. Now, this is very easy to take for granted because for most of us, this has been true for our whole lives, but it's a very recent ability for our species. It only began in the second half of last century. In my postdoctoral fellowship, I worked with Francis Crick, who was the co-discoverer of the structure of DNA. In April of 1953, he and James Watson published a paper in Nature, and in just over a page, they proposed the double helix structure of DNA, explaining how genetic information is stored and copied. Now, there's something about this paper that always makes me tear up when I read it, because the insight changed our world and almost certainly the future of our species. What they realized is that DNA is made of 2 complementary strands wound around each other, and the bases are paired, which just means A on one strand always links with T on the other and C with G. And this gives a mechanism for making a Xerox copy of the whole thing, because you just unwind the 2 strands and then each serves as the template for sticking on new bases in the right spots. But what I want to emphasize is how new this is. 1953 isn't that long ago. World War II was over, Eisenhower was president of the US, and by this point we already had automatic transmission in cars, and color television, and microwave ovens, but we had no idea why your ears look like your father's ears, or your eyes look like your mother's eyes. Just imagine this before the discovery of the DNA code, how difficult it was to understand how inheritance actually happens biologically. People floated all kinds of wacky hypotheses about it, like maybe each sperm cell contained a super tiny embryo, but everyone knew these models didn't work. And even while people were driving cars and watching TV and microwaving, they knew that inheritance was a total unknown in science. And then that all changed with that very short paper that laid the foundation for modern genetics and molecular biology, and ultimately technologies like gene editing. And so that puts us where we are now. For as long as animals have walked this earth, we have been shaped by forces beyond our control, by the slow hand of evolution, by the accidents of mutation, by the blind winnowing of natural selection. The twisting helix of our DNA has been sculpted by nature's chisel. But now, for the first time, we are holding the chisel in our own hands. Just in the last nanosecond of evolutionary time, we now command gene editing technologies, things like CRISPR and single base pair editing tools and epigenetic editing tools and tools yet to be imagined. And these give us the power to rewrite the code of life itself. We can correct genetic disorders, we can eliminate inherited diseases, and we can presumably even enhance ourselves, pushing beyond the biological boundaries that we would have recently assumed are fixed. The rules of genetic inheritance were once immutable, but now they are revisable. So obviously, with this power comes deep questions. If we can edit our genetic destiny, what should we choose to become? Do we cure only what ails us, or do we optimize ourselves, enhancing features like intelligence and strength and longevity? How close are we to doing any of this? Does anything happen to the meaning of human struggle when suffering can be edited away? Will we remain the same species once we begin sculpting ourselves? And who decides? Is it the scientist at the lab bench, the policymakers in government, the parent holding their newborn in their arms? What are the ethical and social and existential questions of putting our genetic future in human hands? So, in today's episode, we're going to step into the frontier of gene editing. What does it mean to be human when we are no longer bound by the limits of our biology? What stories will future generations tell about the choices that we make now? The code of life is no longer written in stone, so what will we write? So I called my friend Trevor Martin, who is the co-founder and CEO of Mammoth Biosciences, which is based here in the San Francisco Bay Area. They are building the next generation of CRISPR products for editing the genome. If you've never heard of CRISPR or aren't sure what it is, hang tight because we'll get to that in a minute. But the quick preview is, how do you build a platform to read and write the code of life? How do you make tools so you get something like a word processor for the genome, where you say, look, I just want to hit Control X to cut a piece of the genome, or Control V to paste it, and Control F to find some sequence inside the genome. So here's my conversation with Trevor Martin. The CRISPR technology is some of the most amazing technology we have, but it wasn't actually invented by humans. It was merely discovered by us. So tell us about that. Tell us about CRISPR.
Yeah, so similar to how we have immune systems, actually, bacteria and small microbes and just tiny little organisms, they have to defend themselves against invasion as well, typically viruses, actually much like us. And one of the ways that they evolved to do this is this thing called CRISPR that has become famous, of course, for its ability to do genetic editing. But fundamentally, nature has used these CRISPR-type systems to protect themselves from viruses, very similar to how our adaptive immune system protects us from viruses. And we've kind of ripped that out of nature and done a ton of engineering on top of it to turn it into these technologies that can do genomic engineering. But that's kind of fundamentally where it came from. And I think it's this beautiful example of leveraging billions of years of evolution and combining that with human ingenuity and a lot of hard work from a lot of scientists.
So let me get straight. So CRISPR, if it's in, let's say, a single-celled organism, a virus injects some DNA and it's got to figure out, hey, that's not mine, and then it cuts it up.
Exactly. So there's a bunch of complicated steps there. First, it has to recognize, hey, that's not mine. Then it has to cut it up. And then actually there's a third step, which is, hey, I should remember this came and I'd want to make sure I protect against it in the future. And it's actually funny. That's where the name CRISPR itself comes from, is that I want to protect against the future. So CRISPR is actually an acronym. It stands for Clustered Regulatory Interspace Short Palindromic Repeats. And this is actually kind of the memory of the cell that the CRISPR systems use to say, hey, these are viruses that have invaded me before, and I want to make sure they don't do it again.
And this happens in unicellular organisms. That's so incredible. Okay, and so you started this company, Mammoth, with the idea to take these CRISPR systems and improve them from what nature has done, or search for other ways of doing it. So give me a sense of how you do that.
Yeah, so probably the most famous CRISPR system is this thing called Cas9. And this was what one of my co-founders, Jennifer Doudna, won the Nobel Prize for a few years ago, was the work in terms of really characterizing and developing this into a genomic engineering technology. And Cas9 was just one example from a certain class of bacteria of this type of CRISPR technology. And one of the fundamental insights of Mammoth is that actually there are all sorts of CRISPR technologies out there, because these are present in all sorts of microorganisms, bacteria, maybe even large viruses, archaea. And our insight was, hey, we should look through all of these alternative versions of CRISPR that are not Cas9 and do a lot of work to develop those. And that could actually have a huge benefit for building genomic medicines, for building diagnostics, for improving agriculture. And that was kind of a fundamental insight that was maybe obvious today, but at the time people were so focused on Cas9 that people weren't really looking beyond that.
So you're looking for things that have already been discovered by nature elsewhere, and you're looking for versions of that do what you want in terms of gene editing.
So the trick there is that you use nature as a starting point, and the unfortunate truth is that when you take these things and you rip them out of nature, actually, usually they don't work at all, but they give you a starting point. And then one of the fundamental insights we had was it's not enough just to go into nature and to say, okay, what other alternative CRISPRs are there? You have to do that, and then you have to do a ton of engineering, and we actually have a whole floor of our building that has these liquid handling robots. They're just running tens of thousands of experiments at a time, and you just kind of grind away, and you can use the latest AI techniques, combining it with the latest in microfluidic handling is what those robots are called. And it's only with that combination of all of that with this kind of usually very wacky kind of natural starting point, that's the secret sauce. Like one or the other is not enough. You have to really have both together. And that was a unique insight as well.
Oh, great. Okay. And so you're looking for these smaller systems, and what's the reason to have them smaller?
Yeah, so one of the giant challenges of the field is how do you deliver these systems to the cells that need it the most? So when you and I think about genetic medicine, what comes to mind? It's going to be things like Alzheimer's, Parkinson's, Huntington's, these really debilitating disorders where they can be basically a death sentence. And it's something that's kind of known in your genome from birth. And the big problem, though, has been what the genetic medicine and the CRISPR field has been focused on is a tiny subset of diseases that are typically not that. And that's amazing for those patients that have diseases that are like blood disorders or like certain liver disorders. And there's amazing progress in those fields. Like, for example, there's now an approved therapy using CRISPR for sickle cell disease and beta thalassemia. Those are overlooked diseases with underrepresented populations. And that's a huge win for the field. But the promise of genetic medicine is not just blood disorders and liver disorders. The promise is to go to any cell in the body and do any kind of edit. And that really is what guides us at Mammoth. And that means you need to go to the muscle, you need to go to the brain, you need to go to the heart. And that has been a gigantic challenge. And it's because Cas9 is actually a big protein. So obviously it's small relative to us, like all proteins, but it's really, really big on a kind of molecular scale. And one of the things that we did is we said, hey, could we create a CRISPR system that's not just a little bit smaller than Cas9, but it's way smaller? And that resulted in a thing we called nano-Cas, nano. And that was really exciting. And there's a lot of skepticism in the field, which is, great for our patents. People are like, oh, these will never work. And it required a ton of work and a lot of these robots doing a lot of work overtime with our scientists. And what's cool though, is that now we actually have data showing that in monkeys, which is a really high bar, it's not, cell lines or mice, we actually get extremely good editing, either equivalent or better than Cas9 with these really, really tiny systems. And these really tiny systems, unlike Cas9, can actually be delivered anywhere in the body. So that's a sea change in terms of what's possible.
And they can be delivered by a virus, for example?
Yeah, so a classic way that you can deliver to muscle or brain, for example, would be with a thing called AAV, which is another acronym that stands for adeno-associated virus. And one of the big limitations of this is that it has a very strict size limit. Like you can think of it as like a semi-trailer truck where you can only fit so much in the back. And Cas9 is just way too big to fit inside it. But these nano-Cas style systems don't just fit, but they actually have a ton of room to spare. Wow. And the room to spare is really important as well, because when you're a scientist, you can start to think really creatively about how do I use that? And one of the key ways that we use that is to fit in the machinery to do different types of edits. So people may have heard of things like base editing or gene writing or epigenetic editing. These are all techniques that take the fundamental CRISPR system and say, hey, what if instead of editing the genome as this like Word document and only being able to delete sentences, what if we could add a paragraph? What if we could spell check a word? What if we could italicize a sentence? These are all kind of different types of edits you can do in the genome, and they require delivery of even more machinery. And that means that Mammoth has really been the only company that's been able to actually deliver not just anywhere in the body, but also deliver any kind of edit anywhere in the body. And I think you have to do both of those if you really want to address all genetic disease.
And any kind of edit means reading or writing.
Yeah, so we also had a lot of work we did on diagnostics as well. And during the pandemic, we actually got emergency use authorization for a CRISPR-based COVID test. So we're super proud of the work we did there. The focus of the company today is very much on the writing side. But definitely, I think there's huge potential on the reading side as well.
Give us an example of the reading side.
Yeah, so basically there, instead of using the CRISPR systems to change the DNA, you can use the CRISPR systems to send out some signal that there's a certain sequence present. So it could say, hey, I found the word potato. And it'll glow green if it finds potato, and it won't show any color if it doesn't. And that's a very powerful kind of concept. And that means you could do really low cost, high accuracy style molecular testing. And that's something that we're very bullish on long term. But as a company, obviously, you have to focus on, certain area, and already, trying to tackle all genetic disease is a limited set of focus to begin with. So that's kind of where we're focusing our efforts at the moment.
Great. So let me come back to something you said. So as far as the writing goes, you can write single base pairs, you can write something longer, you can write whole genes or collections of genes, I assume.
Yeah, you could insert an entire gene. You could kind of change a single base pair out of all the billions of base pairs in your genome. And that's an important philosophical point as well, because I think in biotech, we often get so enamored with technologies. We always think in terms of like, this is a base editing technology, or this is a gene writing technology, this is like double strand break. If you're a patient, you don't care, right? I have a disease, and I don't care how you're doing it. I just want you to cure or treat my disease. And in our case, we can actually cure it. And I think that's where our philosophy is. We'll actually develop many techniques, all the techniques, and actually be able to deliver them. And then for any disease, we might try a couple different ways of doing it. We might say, there's like a way to do it by base editing. There's a way to do it by epigenetic editing. We're not sure which one's going to be the best, but we'll try both and see which one actually works well. And that's a very different philosophy from typical biotech, where you try and create like 10 companies, where each company is doing a different method. And I think that's all fine and well in some ways, maybe also from like how do you maximize like investor involvement in different companies. But from I think, long-term company building and from a patient perspective, I think that's very much like the wrong way to go about it.
Yeah. So tell us about diseases in the brain and what you're thinking of is the future of those. Maybe give me a sense of 3 years, 10 years, where we'll be with that.
Yeah. So to start with a specific example of one that I think kind of is frankly a condemnation of the genetic medicine space is Huntington's disease. So this is a disease that was mapped, oh my God, not just decades ago, like half a century ago, like I think in like the late 80s.
Right.
And it was mapped they mapped it with microsatellites, I believe, on giant gels that were in the lab, right? This is like, not pre-computer.
Nancy Wexler, right?
Yeah, but, very, very early technologies. And we have understood, fundamentally kind of the genetic base of Huntington's for a very long time. And still today, people die from this every single year. And it's a horrible disease where basically, if you have a certain genomic sequence, then, typically in your 30s, you'll have the accumulation of a certain protein and you'll pass away. And it's infuriating, honestly, that we understand the genetic cause of this and we can do nothing about it. So I think the genetic medicine space, and CRISPR in particular, will have arrived and will have really, I think, done a hallmark deliverance of like what the true potential is the day the last Huntington patient dies.
Agreed. Yeah. When is that, do you think?
Yeah, so I think it's definitely within sight. I'm not going to give a specific timeline.
But do you think it's 3 years or 5 or 10?
Yeah, it's probably not 3, but it better not be 10.
Okay.
Yeah. I think in general, like you can kind of see the steps that you need to take, and it's a matter of walking down the path.
Got it. And what has been the problem, given that we have CRISPR-Cas technology and that we have known the gene for Huntington's, it's monogenetic, what has been the holdup?
Yeah, so I think there's a lot of things you have to think about. One is what type of edit are you going to do? Like, can you just knock out the whole Huntington gene, or do you have to think more creatively about modifying it in a more subtle way? And then the second one is, can you actually get the editing machinery to the cells of interest? Can you actually get this into the brain? And those are two of the most obvious problems, I think, that we've really thought deeply about at Mammoth in like the general sense, not just for Huntington's, but for like any brain disease.
Yeah.
And I think that's where the technologies we've developed, like these ultra compact systems and having all these different editing modalities, I think can make a huge difference potentially.
Got it. And so it sounds like you've got can we get it to the right cells? It sounds like you've got a beat on that. But as far as what edit to make, is that something you're experimenting with?
Well, I think that's definitely something where we have a lot of great ideas for any disease about like different types. And that's where we're very unique because we can actually try different methods and we can say, hey, this is our hypothesis, prove it out or not, but not have a hammer and everything has to look like a nail, which is very, very classic, not just in biotech, but deep tech in general. And, the classic startup advice of like find the problem first and then figure out the solution. There's a lot of reasons why that doesn't work in deep tech, right? It's like sometimes you really do have to kind of build the thing and then figure out what the best application is. But that being said, the more you can mitigate that, that's a very powerful idea to get away from hammer, squint at everything until it's a nail. Yeah.
Let me make sure I understand. What's the way that you can test out these different hypotheses? Do you have an animal model of Huntington's?
Yeah, so not speaking specifically about Huntington's, but just generally in terms of different diseases, some diseases you'll have an animal model that's really good. And that means you can actually try it out like in mice or maybe even monkeys and like really get a lot of confidence. For others, honestly, don't have anything. Maybe there's no animal model. So like you have to try it out in cell lines and then kind of make your best guess about what's going to work well. So it's very varied, I'd say, across different diseases and across different tissues. But you want to have as many shots on goal, I think, as possible. Because then when you go to humans, maybe you'll find something surprising. Like you only had cell line work and then it doesn't work in humans. And if you only have one technique, you're kind of out of luck. But if you have like, a backup or a backup to the backup, that means you can actually go in and, do something for patients after that.
Got it. So that gives us a good sense of what the challenge is with something like Huntington's. Now, Huntington's is one gene. If you got it, you're getting Huntington's. But what about other diseases, whether it's Alzheimer's or schizophrenia or whatever, that are polygenic, they involve lots of genes? Does that make the problem exponentially harder?
Yeah, so that's a really, really good question. So kind of building on your point about monogenic disease, so diseases where there's a single gene that causes it, there's, depending on how you count, let's say about 4,000 of those that we kind of are well understood. And that means you have your work cut out for you just in monogenic disease. And, there's a lot of work to be done there. And the one thing I'll mention before moving on to the polygenic is that one of the beauties of the kind of CRISPR technology is that unlike a lot of previous things that have happened in biotech, the first therapy you build with a CRISPR technology is the hardest. And then the second one gets easier, and the third one gets easier, and the fourth one gets easier. And that's very different from a lot of things like, small molecule development, where every drug you kind of go back to the starting board and you're like, okay, well, I got to kind of go through the whole process again. And I haven't, obviously I've learned something, but I'm not going to like shorten the process for the second small molecule, the third small molecule I make. And with CRISPR, that's very, very different because you're using the same technology and you're switching out this thing that's called a guide RNA. You can kind of think of it as like you go to Google and you type into the search engine and the guide RNA is what you're typing. So it's very kind of facile to switch these things out. And I think that means that even though there are 4,000, rare monogenic diseases, I think we have a real shot at tackling them all because the first one's the hardest and it only gets easier from there. And that's very different from classic biotech.
Now, how about the polygenetic diseases?
Right. So on the polygenic side, I think the main challenge there is in an exciting way going to become what to edit. So there are limitations to what we call multiplex editing. Right now, I think the state of the art would be, you could reasonably do maybe 3 to 5 edits in one go, depending on which lab you want to kind of take a cue from. And there's a lot of progress that can be made there, of course. But even for these things where you're trying to edit multiple genes, it's often very unclear. Even if you wanted to edit 5 things and you said, hey, I can go edit 5 things, like which 5 you should edit can be very, very tricky. Like schizophrenia being a classic example, even things like type 2 diabetes. And there, I think there's a lot of progress that could potentially be made in terms of mapping these diseases and really understanding even like, are these edits additive? Like if I edit 5 things, am I getting just the full benefit of every edit? Or maybe is there a sequence of edits that's going to be more beneficial if I do them in a certain kind of cohort together? And these are very complicated statistical questions.
Right.
And it's very non-obvious kind of what the answer is for many of these diseases. And that's where I think there's a lot of additional kind of statistical work that could be done.
Okay, got it. But you've got the technology now to go in there and do these experiments. What do you see as the ethical issues about a society that knows how to edit genes as we move forward? Let's say we're thinking 10 years in the future, 20 years. What do you see as the issues there?
Yeah, well, I think the exciting thing is that we're quickly going to live in a society where we're better at making edits in any cell in the human genome than we are at understanding what to edit, which also has a readout on, your question about the ethics of like, okay, let's move beyond monogenic disease. Let's even move beyond like the classic polygenic diseases like schizophrenia, type 2 diabetes. I think these are relatively non-controversial things where, of course, if you have an ability to cure people, you probably should, or at least I feel very strongly personally that you should. And then it gets into the realm of, I think, people like to think about, oh, well, what about things that are not necessarily diseases, but maybe you want to improve, like whether that's,
your kids' athleticism or intelligence.
Intelligence, these are the classic
height,
ones.
yeah.
And as someone that did a lot of work on the kind of genetic side of the equation, I think one thing that's often lost here is that there's no intelligence gene, just to be clear. Like the gains you can get even from doing a lot of edits on the intelligence side are kind of shockingly minimal.
Agreed. Although if we fast forward 20 years and you figured out, hey, polygenetically, here's some very clever AI way to test this and try that, maybe we'll find out, it's
definitely possible. I think there's a bit of a holy war in terms of, the environmental component versus the innate genetic component.
But the innate genetics doesn't hurt, right?
Right. So let's put that aside for a second and say, okay, let's assume there's been progress made and we have some better understanding maybe of at least what are the best possible edits you could make. I think it's going to be really interesting because if you zoom out, I feel like first of all, this is a question beyond any individual and beyond any company. And it's really kind of a society level question where there's, religious and, ethical and kind of personal and of course corporate kind of viewpoints here. But I think you've seen this in other deep tech areas. You see it with AI right now. Every country is going to have kind of a different view on this. Right. And I think the really interesting thing when you start thinking about human biology, which we all share, of course, is that these decisions very much are not in isolation, right? And it does make you wonder if one country is more willing to kind of go down some of these paths that other countries might find less ethical. Does that create an imperative for other countries just to follow along to stay competitive? And I don't know what the answer to that is, but I think that's the part that seems like it might be most complicated, honestly, is different countries will come to different conclusions. And there's definitely been a lot of work to try and come to like international consensus around these things. But in general, I think that's going to be the trickiest pressure is that even if, let's say in the United States, we make, certain decisions around this is the line, we're not going to do edits for intelligence, but we are going to do edits for anything that's, classified as a disease. Maybe another country decides, hey, actually, I want to give my population supercharging powers to whatever extent I can. And maybe it's a minimal extent, but we're going to try our best. I think that creates a pretty interesting situation geopolitically about like, how do you handle that?
Yeah, agreed. Once we're a species that knows how to modify our code, yes, there'll be the geopolitical things. What do you think it means just in terms of what it means to be a human? How does that change? I mean, let's say we're thinking 50 years in the future here, and you get to choose everything about your kids and perhaps yourself in some ways. But how does that change society?
Yeah, I think it brings to mind one of my favorite movies, Gattaca. I'm sure you've seen it before.
I have seen it, but I thought it was silly in the sense that, just as a reminder to the listeners, your genes predispose you to some particular career in that movie. And so if you have these genes, you're going to be this kind of janitor or whatever, as opposed to the astronaut. And of course, with the nature-nurture debate, that's totally dead because it's always both. Yeah. Tell me why it reminded you of Gattaca.
Yeah, because the part that was most salient to me, I think, was this idea that, let's say one child was kind of quote unquote natural born, the other child was given these certain advantages to whatever degree at birth. And more to the point, being natural born was just a disadvantage. You weren't allowed to apply to be like an astronaut. Like doors were just closed to you, basically. Yeah. And I think that is a very kind of concerning world to live. I don't want to live in that world, in all honesty. And I think you could maybe have like some rationalist argument about why maybe you should do that. But I think just morally and ethically, that feels just really horrible to me. And the whole point of the movie was that actually, no matter how good the science is at that point, there's just certain factors that make that a silly choice. Like the resilience of the individual and their ability to overcome the challenges they had genetically. And I think that's a message that really resonates with me, because I think even 50 years from now, there's going to be many things we don't understand about our biology. And I think if you try and over-rationalize these things and shut doors because like, oh, someone had this genotype and not that one, just inherently you're going to be missing things, and you're not going to be actually as rational as you think.
Well, that's true. But maybe 100 or 200 years from now, we'll be less bad at that. Yeah. It would be, just play devil's advocate, it would be like saying, athletes who are on anabolic steroids won't ever be better than natural athletes, but they will be. They can lift more and so on.
Well, yeah, now there's going to be the enhanced Olympics, I suppose.
Yeah, exactly. And so, but that doesn't affect the career choices like in Gattaca.
I think where I get most squeamish is when it comes down to like the individual choice. Because going on the athletics example, you can choose to take performance enhancing drugs or not, right? And that either shuts or opens doors for you. I think the thing that's more human to me is you don't choose how you're born, right? Yeah. That's a choice that's made for you in a lot of different ways. And I think that's where the type of world that I don't want to live in is where things that are truly outside of your control in terms of like your genome before you're even born determine kind of how you live. And that's not something where I can reconcile personally. Yeah.
I mean, interestingly, we're already in that situation, right? Which is that there's a genetic lottery and you show up in the world with advantages or disadvantages. But now it's just a matter of whether your parents did the right payments and edited.
Well, and I think that's an important point as well, that I don't want to live in a world where there's a massive stratification between the rich and the poor, not just from like a starting point of like, okay, these are the opportunities available to you in terms of schooling, but this is the starting point for you in terms of your genome.
Yeah.
And that's something that we talk about, the inheritance of wealth. Well, you literally inherit your genes.
Yes.
So that's something that could be a very durable advantage in some ways if it's not, something that we think about very deeply before we go down that path, if we go down that path.
So following up on that, can we change features in adults, as in you make it, again, in the future, where you decide, hey, now I want to be like the equivalent of performance-enhancing drugs, and do that at any age?
Yeah, and I think that's kind of where most of the focus is now, is like, well, first of all, for disease, of course, let's say someone was born with Huntington's and now they're in their 20s, like you want to help them out and really, prevent them from having any problems in their 30s. I think for a lot of diseases, one of the key questions you have to answer is like, when do you have to intervene? For some diseases, maybe it could be very early in life. Maybe there's certain biological processes that just take place early in life and you need to do the edit before then, or else maybe you have to come up with some other way of reversing the disease. I think fortunately seems like maybe most diseases you can edit in adulthood, and that can actually have a very material effect on the disease, but that isn't going to necessarily always be true.
Okay, got it. And so coming back to this conversation about what society will become, it may be that some of it is not an issue that you're born with and you have to deal with, but that you make a choice, just like anabolic steroids, that people are doing it that way. Do you suppose that people are going to be looking for things that involve longevity as one of their first aims with this?
Yeah, I mean, obviously there's a huge amount of interest in longevity. I have a personal interest in living a healthy life for a long time anyway. Yep. I think I'm definitely fall more on the spectrum of like health span versus lifespan. If I'm living to be 200, but I'm, decrepit and can't remember my kids, that's not a life I personally want to live. But if I live to a very healthy 110 and where, I die in my sleep, that sounds great to me. Yeah. So I think longevity is definitely an area where gene editing could play a huge role in terms of, what are the processes? Is it like, some sort of mutation accumulation in certain areas that's like causing these cells to age in the way they are? And could we reverse that with genetic editing approaches? I think that is a very reasonable and potentially promising line of research. I think that in general, some of the biggest longevity things we can do though today is like, heart disease, cancer obviously being a big one. And those are of course directly addressable by genetic editing as well. And it goes to interesting questions of like, should we edit millions of people in the US to reduce their risk of heart disease by 30%? That could save tons and tons of lives. It's a relatively large intervention for them. So obviously you'd have questions about safety. So far, CRISPR seems to be very, very safe. at least based on the trials in humans so far. So that's something we could do. Is that something we want to do as a society?
Why? What would be the pros and cons?
So the pro would be you reduce, let's say, heart attacks and all-cause mortality, essentially, in a huge segment of the population. The con would be there's going to be some expense associated with that. Do we want to pay that expense as a society? Maybe it saves us money in the long term because both people are more productive and you're also spending less in kind of end-stage care in the hospital.
So you see that as a government program that's paid for as opposed to individuals saying, hey, I'm going to pay for this.
Well, I think that's a very interesting question as well. And I think you could see stratification there of maybe, frankly, wealthier individuals choosing to get a lot of these types of technologies, whereas, is that available to everyone? That's a kind of choice for society. And then, of course, the big one would be safety. You only want to do that if it's extremely safe, right? Because you're trying to prevent disease kind of in aggregate. So it needs to be extraordinarily safe for each individual.
Do you think, if you're interested in longevity, do you think you're going to see this in your lifetime where there are edits that can be made, let's say 40 years from now, where you say, hey, this is great, I'm going to live longer, I'm going to expand this?
Yeah, it's an interesting question. I wouldn't be surprised if there's things that can maybe get you 10, 20% further along. Are we going to see like a doubling? I mean, that'd be great. Sure. I'm not going to be root against it. I would be surprised, frankly. We've run a natural experiment. There's billions of humans on Earth with all sorts of genomes. No one seems to live over 110, 120, let's say. Yeah. That doesn't mean, it's not possible, but I think that one of the areas where I think we'll see way more progress on is like health span within that period.
Yeah.
So like if you can live to an extraordinarily healthy 110 where you're hiking mountains and you're having fun with your kids, that's already a huge extension of that. That's like a twofold extension in life relative to what a lot of people really kind of practically experience. And I think GLP-1s and things like that, the obesity drugs from Eli Lilly and Novo and others, I think that's also kind of shown that there can be these kind of giant mass markets in biotech. And longevity is the other one that everyone always kind of thinks about. But there's others as well. You can imagine things that mitigate like alcohol consumption or, other areas. I don't think GLP-1s are going to be the only kind of $1 trillion drug in biotech, but I think it's the first. And I think hopefully that kind of inspires everyone else in biotech, including us at Mammoth, to really think more broadly and more fundamentally about, okay, what are the things we can do that actually change society for the better overall, not just necessarily for specific diseases. You got to start with specific things and knock out rare diseases. But I think in terms of like the long-term vision of Mammoth, I think that becomes very, very exciting.
And I've been throwing out numbers like 3 years, 5 years, 10. But what do you see, given your incredibly specific view on what is happening right now, what you're capable of doing? What is that timescale that we're talking about?
Yeah, so I mean, going back to the beginning of the conversation, already today people are being treated for sickle cell disease using CRISPR. So that's today. I think over the next 5 years, for the tissues that are kind of most easily accessible, like blood and liver, people are going to be shocked at how much progress is made at curing rare diseases in those areas. And these are kind of the things where it's like it happens slowly and then it happens all at once. Yes. Like it's very much like that. And then I think over the next 10 years, you're going to, because of Mammoth, be able to see us knocking out diseases in like muscle and brain and all the other tissues of the body. And I think it's going to take time and there's a lot of diseases to go through. But I think if we're sitting here a couple decades from now and people are still suffering from rare disease, we've done something incredibly wrong. Like things have gone off the rails somewhere. And that's insane. Like to live in a world where genetic disease is really a thing of the past would be transformative, both for those patients, but also I think for society generally. And I think once you've done that, that's when you, in parallel, because now you're showing the safety, now you're showing that this is an effective technology, I think you can start to dream even bigger. You can start to think about like, how do you go after reducing cardiovascular risk? How do you go after even more esoteric things like thinking about health span? Like there's certain people that have short sleep and they actually only need to sleep like 3 to 4 hours a night. Nice. Like what if we could all do that?
Right.
Like just really thinking about how do we transform society for the better in kind of more aggregate ways. That's really long-term where I get very excited.
Unlike traditional medicine, which addresses symptoms of disease, this gene editing technology in general could change really fundamental aspects of who we are. Mm-hmm. And so what do you think this does to personal identity?
Yeah, I think to your point, this is a very different way of doing medicine. Like often medicine is treating symptoms, often very effectively, but really treating symptoms. Whereas this is going into the core, into your DNA and really solving the problem at its
Right, but
foundation.
I mean not just helping with disease, but enhancing humans, and changing who we are. So yeah, what does that do to our sense of personal identity?
Yeah, I think it's definitely something where we have a deep kind of personal identity, obviously, in terms of how we think, how we act, how we dress. And I think long-term, if we're just truly thinking like 100 years here, maybe we start to think more of our genomes as our clothing, right? Like we think of our clothing as representing our personal identity, and that's fine. And that's something people embrace, and people have all sorts of different styles and things they do. But right now, that's not very much how we think of our genomes. But if genomes become malleable and they become something where you really do have this option of kind of choosing what things you want to keep or change, maybe we start to see that more as kind of extension of ourselves that's not core in the sense of it can never change, but core in the sense of I want that to reflect who I am as an individual, and I have the choice of how that looks.
Oh, that's great. Yeah, in the same way that someone might go off and get blonde hair for a while and red hair and dark hair and change the color of their eyes, whatever. There are ways of doing the stuff now, and there's going to be better ways or more direct ways of doing it. What do you suppose that means in terms of motivation and earning something? Like, for example, I go to the gym and I lift weights and it's really hard, but if I could go and
Sure, you get your myostatin gene blasted and you just have huge muscles from sitting home on the couch. Yeah. I think that's an interesting question at the society level, because why do we care about muscles? Why do we care about other things? Like at other times in society, having pale skin has been something that people value. I mean, still today in many places. And it's because, you don't have to go work in the farms. That means you're a high class individual with a lot of money. Or in some societies, even today and in the past, being very overweight was a sign of high status because you're not starving. And I think that what that could mean, and this is just like, purely hypothesizing, is maybe being healthy remains an important thing, but it's not a reflector of status in the same way it is today, which would be awesome. Like what if actually everyone is walking around super healthy? Yeah. Amazing. But maybe that means that there's less of a status differentiator of, this person has time to go to the gym and they work very hard. So something else, but we're humans, right? We love, frankly, it seems to stratify ourselves based on status. So some new thing will come up and it'll be like, they have a blue hat. That's the sign of, high status. And we could pass a judgment on whether that's good or bad. But I think that's probably what would happen, is that it just means that society's better off overall because everyone's very healthy and has the right amount of muscle. But there's a different status game being played in terms of, when you go to the club on a Friday or whatever.
That was Trevor Martin, co-founder and CEO of Mammoth Biosciences. We talked about the emerging tools that allow us to edit life at its most fundamental level. The conversation and the tools he's building, this allows us to look at the near future in which we can slice out chunks of the genome or rewrite individual letters or even fine-tune the expression of genes without altering the sequence. And eventually, this gives us finer and finer control over our biological destiny in ways that we're only beginning to understand. Because these technologies are not going to only give us the chance to fix mutations, they're going to expand what is possible. They will redefine the relationship between who we are and who we might become. Where we stand now is in a field of question marks that no single thinker can answer alone. Questions about the line between therapy and enhancement, between responsibility and hubris, between embracing the malleability of life and respecting everything we don't understand about it. Only one thing seems certain. At this point, we are no longer just passengers on evolution's very long and winding road. We are taking the wheel. And with that power comes a great responsibility, the weight of choices that are going to ripple through generations, the weight of choices that will shape the genetic landscape of those who come after us. In the end, our role is going to be to learn how to annotate the book of life with great care, to correct its most tragic errors while preserving the poetry of its imperfections. We now see the genome not as a finished book, but instead as a draft in progress. And that compels us to constantly ask ourselves, how much of the story should we change? What kind of story do we want to tell? And how can we be the most careful custodians of the possibilities? Go to eagleman.com/podcast for more information and to find further reading. Send me an email at podcast@eagleman.com with questions or discussion. And check out and subscribe to Inner Cosmos on YouTube for videos of each episode and to leave comments. Until next time, I'm David Eagleman, and this is Inner Cosmos.